Welcome to my world, the world of the medical laboratory scientist. Health is wealth, knowledge is power. The topic is epigenetic effect on hereditary disease. What we shall be studying is the sickle cell disease. When we talk of epigenetics, we are more or less talking about the environment factor, the factors in the environment that affects the gene expressing itself, the gene expression. Because at times you have hereditary disease and you find that some people don't come down with that hereditary disease, even though they have the gene composition in them. But epigenetics has more or less now exposed, so that with epigenetics these effects could quickly come out, and it may also prevent it in a way. A study on sickle cell disease that tried to look at the effect of certain drugs and even blood transfusion on the aging process, how the sickle cell disease patients age. The finding in that study shows that the aging process is sped up in with certain drugs, and with even blood transfusion it is lowered. And these are ways which things I shall be exposing in the slide session. We can come to understand epigenetics very well from just assuming or accepting that once you have this hereditary disease, you are bound to come down with it. No, epigenetics plays a very strong factor. And you can even understand epigenetics when you look at the longevity that is so stratified country by country, because the condition, the environment in different countries creates that longevity for them. It's very easy to assume that we know what is causing it. That some people in the tropical countries, the least developed economic disadvantaged country, they have shorter lifespan, while those who are in developed countries are having longer lifespan. But epigenetics will do more than just assumptions. It will go really into studying how the genes are manipulated to now create that expression or prevent the expression of the mutated gene. Now, I've always been saying, if you like my presentation, press the like button, subscribe, and more of this information will be exposed to you in the slide session. The outline: objective introduction, hereditary diseases, epigenetics effect, laboratory diagnosis, and sickle cell disease cure. The objective: to introduce epigenetic effects in elongating the lifespan of carriers of hereditary disease in their genes, with suggestion on what laboratory parameter to evaluate in people having hereditary diseases genome, and expose the successful cure of sickle cell disease in a 12-year-old boy. The introduction: epigenetics involves changes in genes expression that do not alter the DNA sequence. In sickle cell anemia, epigenetic modification can influence the severity and frequency of disease episodes, which is the cause of the many complications, stress associated with the condition. The modification does not affect the gene sequence, but it merely acts as an on and off switch on the DNA and is influenced by environment, diet, and aging. The key types of modifications are DNA methylation to the start of the gene, thereby silencing it; histone modification to ensure that it is very tight and difficult to loosen. The third is microRNA, small RNA that can also regulate gene expression. Hereditary diseases are diseases that are genetically passed down to other generations of a family. Hereditary diseases fall into three main types, namely the monogenic, which is the single gene mutation, where it is only one single gene that is mutated, like in cystic fibrosis or sickle cell. There is the chromosomal changes, where the whole chromosome, where you probably have more than, especially with the X-linked chromosome. Sex-linked chromosome, you have probably extra Y or extra X. Then there is the multifactorial chromosome, which has to do with gene-environment mix, like heart diseases, diabetes, many cancers, plus rare mitochondrial diseases. These are mutations, genetic inheritance that are passed through the mothers. They stem from inherited DNA errors affecting single genes, entire chromosome, or gene-environment interaction, leading to diverse health issues. Sickle cell anemia is a genetic disorder caused by a mutation in the hemoglobin gene, leading to the production of. Talking more about hereditary disease, we have the autosomal dominant, where the gene that is really dominant takes effect, one copy, and then we have autosomal recessive, that is the recessive gene that really brings out the character, the phenotype that you see. X-linked dominant and recessive genes are on the X chromosome, affecting male and female differently. Color example is the color blindness. Mitochondrial inheritance, just as mentioned, passed from mothers through mitochondrial DNA. Now to the epigenetic effect. When we talk about effect, it's like trying to name the cause, the causal agents. So the epigenetic itself, where there are certain things it causes, and one of them is now discovered to be it could help in longevity, that is increasing lifespans. Let's take a conclusion from a study, current study. The current study is the first, to our knowledge, to examine the effect of hydroxyurea and blood transfusion. These are agents used for sickle cell disease management treatment. Participants showed a 19% faster aging rate, with hydroxyurea increasing and blood transfusion modestly reducing the pace of aging. So that shows the effect of hydroxyurea on the sickle cell disease patients aging quickly, even though the drug itself helps in treatment. Hydroxyurea treatment is associated with a higher pace of aging and shorter telomerase length. When we talk about telomerase, we are talking about what has been discovered, that this chromosome, the extension of the gene, the basal end, as shown in that picture, will now be lengthened, which helps more or less to say one's lifespan is lengthened. The shorter the telomerase, the shorter the lifespan. Whereas blood transfusion show a modest reduction in the pace of aging and longer telomerase. These findings highlight the differential impact of sickle cell disease on epigenetic aging markers. Understanding this relationship can guide personalized therapeutic strategy to mitigate premature aging and improve the quality of life in sickle cell disease. The laboratory diagnosis. The laboratory can help identify those carrying mutant genes that are hereditary, and use it as a predictor of health conditions or challenges that may occur. With yearly study, it reassures one of perfect health or what to do next. Epigenetic testing offers a powerful tools for predicting disease risks by analyzing how environmental and lifestyle factors impact gene expressions. This testing modify risk assessment for chronic diseases, helping doctors and researchers better understand individual vulnerability. Tests include DNA methylation testing. For example, in breast cancer, the DNA methylation changes can indicate early stage risk. Heart diseases. Epigenetic profile help track progression and predict outcome. The metabolic disorder too. Specific markers provide insights into conditions like diabetes and obesity. The samples that are usually used are blood, the buccal cells, the saliva, and any cells that could be studied in terms, because all cells contain these genes. Probably What we are talking about is in almost all the cells. You could just single out the red blood cells that have no nucleus. So without a nucleus, the only thing that may possibly be there is the mitochondria. Now finally, the SCD cure that has been discovered. The current treatment for sickle cell disease anemia include the hydroxyurea, L-glutamine, crizanlizumab, and voxelector. And you can include blood transfusion, which reduce complications and improve quality of life of these patients, the sickle cell carriers, sickle cell disease condition. However, there's recent discovery: a 12-year-old boy from Indiana is getting a new lease of life after a stem cell transplant cure his blood disorder. They cure this blood, the SCD. So let's read the story as exposed. Since he was born, Elliot Petty has struggled with sickle cell disease, a group of inherited blood disorder that causes a shortage of healthy red blood cells. The condition, which can lead to frequent infections, pain, delayed growth, and vision problem, caused Elliot to miss out on activities that other kids his age were enjoying. When that's his statement, was younger, I played sport a lot, but then I got too cold from my sickle cell, Elliot told the television. Yeah, continuing, that's why I started quitting all my sports, my baseball and soccer, Elliot said as he sat on his back porch next to his family. He just couldn't enjoy life as a kid. His mom, Trace Marshall, added. But a year after receiving a stem cell transplant at Riley Hospital for Children at Indiana University Health, doctors now say Elliot is free of the disease. It was a huge sigh of relief, the hospital said. We shed some tears for sure. According to the Center for Disease Control, bone marrow and stem cell transplant are the only cure for sickle cell disease. But the procedure comes with risks and can present serious side effects. They can even result in death. That's the warning. So, like I've always said, my effort is to create more understanding, create knowledge, awareness, so that we can enjoy life in full. This young man, this young boy, 12 years old, is now enjoying life. You can imagine what health condition has really caused the boy. So the more knowledge we acquire, the better for us. This will be the 212th video that I am releasing. So you can watch many of my videos on this channel. Thank you.